know if anything is not clear. So I want to just talk about essentially hybridization and admixture and introgression and genomic approaches to assessing those things. And I don't promise to give sort of an exhaustive view of those things, but I want to go into a few of the basic analyses that you can do with the types of genomic data that we've all been talking about in cases where you have hybridization and introgression. And particularly, I want to focus on cases where it might be of conservation concern and you're interested in, for instance, introgression from a common introduced invasive species into, say, a rare threatened species. So just to think a little bit, some definitions first. So these words hybridization and admixture and introgression get thrown around and treated in some cases as if they're synonymous. They certainly have plenty of overlap, but there are some important distinctions. I don't present these as the best possible definition or even very good definitions of these terms. I'm sure you can come up with better ones. But I think of hybridization as simply the process of interbreeding between individuals. And I say between two different groups, parentheses, species. So it's often simplest to think of hybridizations happening between two different species. But really, we can think of it between two populations or two groups, however we would want to define those groups. And hopefully those groups have some biological reality, how we're defining them. Admixture is essentially the result of hybridization. You get genetic mixing between groups. And you can talk about admixture at a population level. So an admixed population is one that has at least a history of hybridization. Or at the individual level, you can think of an individual that has ancestry from, say, two different species is an admixed individual. And then introgression, I think of as the flow of alleles from one species into another. So again, the result of this sort of hybridization process, but specifically looking at particular loci and thinking about alleles at those loci moving preferentially from one species into the other. Okay, so just sort of visualize the genomic aspects of these things. Suppose you have these two species, and they each have a genome, and they're differentiated to some extent. The genomes are differentiated to some extent. If you have hybridization between them, you start to get genomes that are mixed. At the simplest level, at the F1 stage, you would get something that's heterozygous at all loci where those two species differ. And if that process continues and you start to, for instance, have back crosses and later generations of hybridization, you start to get a whole mix of individuals with ancestry that comes from both parents. And you would presumably, if you could look at the genomes of all of these individuals, you could see that mixed ancestry spread out across the genome. So what this means is that essentially genetic information, alleles, are moving around back and forth. And with generations of back crossing and interbreeding, alleles and genetic information can move back and forth between these species. And in particular, this sort of situation is most of interest when the species don't just sort of collapse together into one big mass, but they remain distinct. So these species remain distinct and keep their own sort of genetic identity or ecological function or ecological distinction or whatever. But nonetheless, there's some hybridization going on, so the genetic material is flowing back. Okay. So obviously, since genomes are complex things, this is not going to be a simple picture. And we can think of, at different points along the genomes of these organisms, different types of processes going on. So in some parts of the genome, we might have alleles at particular loci sort of moving back and forth as a result of hybridization. At other parts of the genome, we might not find that. And one way in which people are interested in looking at these sort of genomic patterns of hybridization is to understand what loci are actually responsible for maintaining reproductive isolation between species. And so one approach is to basically look for the parts of the genome where you don't have alleles moving back and forth, and use that as a way to map loci that are responsible for keeping those species distinct from each other. Then you can also imagine areas of introgression, as I say, that can be in one direction or another. So for instance, alleles from this species might preferentially, through this hybridization and backcrossing process, alleles from here might move into this species. You may even at the same time have another part of the genome where alleles from this species tend to move into that species. So across the genome, you can have these different sorts of processes. And so this can lead to things like adaptive introgression. I'll show a couple examples where these are still two distinct species or two distinct groups, but there's some genetic information that has crossed over as a result of hybridization and is now favored by selection in this species. So it may, for instance, sweep across the whole range of this species because it's favored by selection, even though its origin is hybridization. Another way that can happen is if, as I'll talk about, if this species is, for instance, an invasive or an introduced species, and this is a native one, particularly one that we might be concerned about from a conservation perspective, genetic material that introgresses in this direction and then increases rapidly in frequency over here because of selection, we can think of as a super invasive allele. So something that is rapidly moving into the native population's genome. Okay. So here's just another way of thinking about these same sorts of things. If you imagine, here these are like little phylogenies, time is moving down. And these colored lines represent the genealogies of different haplotypes or different alleles. And you can have various scenarios for how these species down at the bottom might end up with essentially shared polymorphism. And it could be that the polymorphism traces back to a common ancestor, but it could also be an introgression event like this, where there's some hybridization between these two species, and this A2 allele, in this case, sort of crosses over to this other lineage. So there are lots of cool examples of this, and in particular, all these genomic techniques that we've been talking about have allowed people to really get into detecting these cases. So this is a paper from a couple of years ago looking at cichlid fish in Africa. This was rad data. Essentially showing hybridization and hybridization leading to, if not new species, then at least new populations, new well-defined populations. And because of the genomic scale of data that they had in this study, they were able to track this sort of genome-wide picture, which gives you this tree, but then a few particular loci that introgressed as a result of hybridization and were adaptive. And in particular, those loci that show those pathways of introgression are linked to male color pattern, which is a big ecologically and sexually important trait in these taxa. Here's another really cool example of these heliconius butterflies that you may have heard of. It's this big species complex. They have all sorts of interesting color patterns. And in particular, they have color patterns that exhibit mimicry. So one species will be nasty for predators to eat, and another species will look just like that first one so that predators don't eat the second one also. Some of those color patterns and some of those mimicry color patterns are the result of adaptive introgression, where there's hybridization and alleles at the loci that determine those different color patterns have popped over from one species to another and increased in frequency because of selection. So all of these complex color patterns are largely the result, or in part the result, of adaptive introgression because of hybridization. Okay, here's another example. This is introgression from Neanderthals into humans. Most modern humans have the great benefit of having some small part of Neanderthal ancestry as a result of ancient hybridization and adaptive introgression. So here's just looking across part of chromosome 9. These little spikes are places where there's Neanderthal ancestry showing up. Okay, like I said, lots of examples. Here's another example. Introgression of essentially pesticide resistance from this species down here into the species up here. The yellow shows the frequency. And if you scan across the genome, so the yellow here, this block, is essentially the introgressed part of the genome that has moved from this species into populations of this species. So again, this is an adaptive introgression sort of scenario. And you can imagine how you would be able to detect that kind of thing with fine-scale genomic data. Okay, so like I said, this also has conservation implications. And so there are a lot of issues with hybridization as essentially a threat to native biodiversity. So if you have species of conservation concern that are hybridizing with other species and receiving genetic material that's introgressing into the species of conservation concern, that introgression could result in some consequences, like the erosion of locally adapted phenotypes, outbreeding depression, which is essentially a reduction in fitness as a result of breeding between groups. This term genomic extinction, the idea that essentially with enough hybridization and introgression, you lose the remnants of the genome that used to distinguish that species of concern. So it becomes sort of diluted or swamped by introgression from the hybridizing species. And then finally, there are legal issues. Essentially, when we write conservation laws, we have to be specific and define what we mean by a species or a population or all those sorts of things. And those laws have a hard time dealing with.
We're just going to unmute the mic up there.
Oh, okay. Sorry, Joanna. So we have legal issues that essentially it can be hard to deal with. How do we define these hybrid taxa, these hybridized populations, and what do we do with them? Okay, so here's another example. This is red wolves. This is the coast of North Carolina. So red wolves are a very rare species that were actually a captive population, and they've been reintroduced. But there's a big problem with hybridization between red wolves and coyotes. So coyotes, which are actually not historically native to this area, are now pretty common, and they hybridize with these red wolves. So this graph, if you can see, the white bars are red wolf litters, the black bars are hybrid litters. And there's actually active management to try and reduce this hybridization and reduce the introgression of coyote alleles into the red wolf population. But if you look across individuals, here are a bunch of individuals, and it shows the estimated proportion of ancestry. This is using, let's see, 17 microsatellite loci. The black is red wolf ancestry, the really light gray up here is coyote ancestry. So you can see there's a mix of individuals. This one looks like an F1 because it's basically half and half. This one is maybe a back cross. It looks like just about 3/4 and 1/4. But you can see there's a mix of ancestry. So this red wolf population that's out there now is a little bit hard to define what the genomic makeup of that population is. Okay. Any questions on those sort of empirical examples? So there's lots of interesting things going on there. I want to talk about just a few sort of analytical approaches that we can use to think about quantifying these things, and in particular to look for particular loci that show signs of elevated introgression. Okay, so these are 4 of the sort of analyses that I want to just mention. So one is basically thinking about allele frequencies. So if you have some hybridized population, depending on the degree, the overall levels of ancestry from the 2 different species, you have some expectation about the frequencies of alleles at loci where there's, different alleles from the 2 different parents. And outliers from that distribution might indicate the effect of selection. So here's a study of salamanders in California. So the native California tiger salamander interbreeds with a related invasive species. Here, just scanning across, I think, 64 different markers across the genome, what's shown is the introduced allele frequency. So the frequency of the allele from the introduced species in these hybridized populations. And you can see at most of these markers, it sort of bounces around. These are different populations. So there's some variation, but there's sort of a trend. But then a few of these markers that are in red that you probably can't see, a few of these markers have much higher frequency of the introduced species allele. And those markers share that characteristic across multiple populations. So this is pretty good evidence that these loci, or loci close to those markers, are somehow favored by selection in these hybridized populations. They're increasing in frequency, and so they're showing much higher levels of introgression. So this is just that same data set. So each of these little bars is a population. Is that right? Yeah. And showing different allele frequencies. The white is the native species, and the black is the introduced species. Across all of the markers, the median allele frequency is shown here. So this is roughly the average. These all look like essentially the native species. But at those 3 markers that I showed you on the last figure, the allele frequency of the introduced allele is much, much, much higher. And it's basically sort of swept across these populations. Okay, so this is a relatively simple idea. Look at outliers, just to sort of hammer it into your brains. Some really simple simulations. So imagine you start with a hybridized population that has 50% makeup of 2 different species. If you look at just one locus and think about the change in allele frequency over time, say you have these 2 species come together in equal proportions back here, and then you track allele frequency over time. After 200 generations, if you have a population of 1,000, the allele frequency might look something like this. How many people have seen figures like this? This is the classic standard genetic drift figure, right? This is plain old genetic drift. I'm just setting it in the context of imagining that this 50-50 allele frequency is the result of hybridization. So you expect it to stay the same if it's neutral. But since the population is finite, you have genetic drift. And the allele frequency over time bounces around. Now we can think about what happens at, say, other loci. If you have a completely independent locus somewhere else in the genome that also starts at a frequency of 50-50 over here, it bounces around independently, and it does that. So again, this is classic genetic drift. If we keep adding more and more and more and more and more loci, we can start to see the pattern. So all of these are behaving neutrally, but they're still changing over time in allele frequency. Another way to look at this, now I'm going to show you, imagine if we take a cross section at a particular point in time. And we're just interested in the distribution of allele frequencies across all of these independent loci. So I'll just do that. So after one generation, if we start out with a bunch of loci that are all at allele frequency of 0.5, after one generation with genetic drift, they're going to start to spread out. And then, after 2 generations, they spread out a little more. 3 and 4 and 5. All right, who can write down the equation for that distribution? Who can tell me what kind of distribution that is? There. I hear lots of mumbling. I don't understand any words. Beta? 1 over 2n is proportional to what?
Either not a drift per generation.
The variance in this distribution per generation. So what kind of distribution is this? It's essentially a normal distribution, or binomial.
That's only true because it started at 0.5.
True. Excellent. Yes, and if I
started with 0.9 and 0.1 and be highly skewed.
Yes. Should we do that? For the fun of it? Here, start at frequency of 0.1. This is a closed population. Except a big population, so this is going to take a while. Eventually, things are going to pile up at 0, right? Because some of these loci are going to get fixed. Here's a little population genetics 101 question for you. If I started this at a frequency of 0.1, and I run it until every loci, every locus is either fixed over here or fixed over here, how many will be over here versus over here?
The frequency of it, we always see the same. Among all the populations, 90% go to fixation for 0.9 and 10%. Assuming it's just drift.
That's right. So if I did that, the proportion of loci that end up here versus here would equal the starting allele frequency. So again, this is all just classic quantitative or population genetics, genetic drift, principles of genetic drift. But the point is that things don't stay the same over time. They change, but they change in a way that we can describe this sort of distribution. And under a really, really simple model like this, we can even write down an equation for what the variance and skew and everything of that distribution should be. So we can write down, if it's a simple situation, we can write down a null model for what the distribution should look like across loci given neutrality. So then what does that let us do? If we can write down our neutral expectation, we can test for selection. Right. So we can compare against the neutral distribution to test for selection, and that's essentially what we're gonna be doing. Okay. So when we get to the little hands-on exercise, we're going to be doing this simple test that comes from this paper, Jeffrey Long in 1991, which is a little bit more complicated than that really simple scenario that I showed you, but not by much. Essentially, this is just a matter of estimating the variance in that distribution across loci, and then testing whether there are loci that are significantly beyond what we would expect of that distribution under neutrality. Okay. So one thing that makes it a little more complicated is in a test like this, we don't actually know what the population size is. So we have to essentially estimate it from the observed distribution, but nonetheless we can statistically identify outliers. So that's what we're going to be doing in just a little bit with a couple lines of our code. Okay. Another analysis that we can do is this sort of genomic cline analysis. And here are just a couple papers that both of these papers actually produced R packages that you can go online and download and run in R. So this one's called Introgress, this one is called HIS. So the basic idea of a genomic cline is that we go back to this sort of scenario, and suppose we arrange all of these individuals in terms of how much ancestry they have from one species versus how much they have from the other. So the pure species 1 is over here, and pure species 2 is over here. The frequency of, for instance, the frequency of an allele that comes from species 1 should be 1 here and 0 here, right? And somewhere in the middle for all these hybridized individuals. That makes sense? So the idea of a genomic cline analysis is that we expect alleles that are characteristic or diagnostic for one of the other species to show this general pattern. But if there are alleles that are preferentially flowing from introgressing from this species into this one, or this one into this one, that curve like this will shift. It might shift this way or shift that way. So that curve, that's what these are, essentially. We can represent, here you see this x-axis is called genome-wide average ancestry. So just like this picture, if we arrange all of our individuals according to their genome-wide proportion of ancestry from one or the other parental species, then for any locus we can plot a curve that looks like this, that shows the allele frequencies of those loci as a function of genome-wide ancestry. Okay, so this, these figures, how many of you have heard of cline analysis in terms of ecological space? So that's where it is sort of more commonly used, and that's what this top one is referring to. It's a spatial position here. But we can think of it in terms of not spatial position, but sort of position along this genome-wide ancestry axis. But that doesn't make sense. Let me get into some of these. These are two different ways of representing these genomic clines. You can see there are these three alleles here where this curve is shifted way over. Those are the three loci that popped out before. This is another way of showing it. Those are the three loci that are introgressing much more rapidly than you would expect, much more than you would expect under neutrality. And so the curve of these genomic clines is shifted way over, showing the same sort of pattern. Show a couple other examples. So this is one of the other analyses for looking at clines. And in this analysis, you could look at not just allele frequencies, but actually genotype frequencies. So looking at frequencies of homozygotes and heterozygotes. And so on all of these, there's the light gray and dark gray patches. The dark gray is the frequency of homozygotes from this species. The light gray is the expected frequency of heterozygotes. And so by looking at how, say, an empirical data set differs from the neutral expectation for these things, you can conclude all sorts of different things about selection on particular loci. Whether it's things like underdominance, overdominance, directional selection, all sorts of things. So here's an example of that sort of analysis. This is in sculpins in Europe, a couple different hybrid zones. Each dot on here is an individual. And you can see all of these individuals and each plot is a locus. You can see all of these individuals are either homozygous for one, a heterozygote, or homozygous for the other allele. And if you fit a curve to these sort of data, the solid line and the dashed line are the empirical data, and the light gray and dark gray are the neutral expectation. And you can see, for instance, best example, here you can see the neutral expectation goes up like this. The empirical data is this dark line that goes up like this, so it's much lower. It's shifted that way. So it looks like there's essentially introgression in this direction, from this species to this species, at that local. Here's one where you have, and this one where you have a real deficit of heterozygotes compared to what you would expect. Does that make sense? So what the nice thing about this sort of cline analysis compared to the allele frequency thing that I was just talking about is that you can tease out essentially multiple different signatures of things. It's not just a single aspect of allele frequency. It's shifts in these different curves, and they can shift left or right, or get flatter or steeper. And those different deviations from the neutral expectation can tell you different things about what's going on. Here's another one. How many of you have heard of the ABBA test? Fun to say. So the basic idea here is to test for introgression between two species when you have information on essentially outgroup taxa in a phylogenetic context. So this is getting back to that same heliconius butterfly example that I showed before. If these are the two species you're interested in, and you're looking at whether there is introgression from this third one, you can ask whether there's introgression, a signature of introgression from this one into here, or this one into here. And the way you do that is, if you have this fourth out through taxaway down here, you can tell what the ancestral allele is at a particular locus. And then you can, and if there's a change that happened on this branch of this tree. So these alleles are labeled A and B, which is where the name comes from. So here's the ancestral allele. Here's the derived one. If these two differ in which version they have, the ancestral or the derived, A or B, you can ask across loci, is there a preference in whether this one has the ancestral one or this one has the ancestral one? And that the derived one, the B allele, is an indication of introgression from the species into one of those. So you can test whether loci differ from the neutral expectation. So here in this figure scanning across the genome, we have the genome-wide estimate of this statistic, and outliers above or below. No, this is a null expectation. These outliers are the signature of what seems to be adaptive introgression. Okay. Here's another way of looking at it, and this is why this butterfly system is so cool, because they've narrowed down these regions of ABBA sites and BABA sites, and where they are. And as I said, it's the genes that control these wing color patterns. One last sort of way of looking at these sorts of data. If you have a plot like this with individual level of admixture on the x-axis, so from pure species 1 to pure species 2, hybrids in the middle, and then here you have heterozygosity, so the proportion of loci that are heterozygous. After one generation, you can see how this picture will change over time after a hybridization event. So after one generation, you're going to have pure parents and F1s. And the F1s will be purely heterozygotes and have a 50-50 proportion of ancestry. After the second generation and third and fourth, and so on, those patterns of heterozygosity and hybrid ancestry will shift. And so this is a way to essentially estimate the age of a hybridization event. So if you had two species that hybridized and formed some admixed population, the distribution of admixture and heterozygosity tells you something about how long ago that happened. So again, this is applied to some empirical data. This is back to the tiger salamanders. And these different populations show very different pictures of this relationship between admixture and heterozygosity, indicating essentially different histories of hybridization and different timings of those hybridization events. Okay, any questions on that stuff?
Well, presumably they've eliminated all selection from these things to see these patterns. Are these all neutral loci?
These are, the short answer is nothing's neutral.
Right.
The long answer is I'm not sure if they actually did eliminate them or not. But presumably you could look at outliers from this sort of distribution.
What's the best way to initially determine, or I guess the first step to see if the introgression you're seeing is adaptive or is going to have detrimental consequences? How do you approach that?
Well, you could try and measure fitness as a function of genotype at a particular locus for function of hybrid ancestry.
So like what parameter of fitness?
Well, so fitness is a notoriously hard thing to measure in a lot of cases. Depending on what organism you're looking at, you can measure survival or reproductive output or lifetime fitness. Yeah. Okay, so we're going to get into this sort of hands-on exercise using the long statistical test. And I'll just introduce the data that go into that. So this is a collaboration that I've been working with Gordon and lots of other folks here on for a little while. We're looking at introgression in westslope cutthroat trout. So this species is native to the area around here. A very nice map, but we are in here somewhere. Rainbow trout have been introduced. They're one of the most widely introduced species around the world. They're dumped all over the place for fishing. And rainbow trout have hybridized with the native westslope cutthroat trout. So here's right back to your question. Here are direct measurements of fitness, showing that hybrids are actually less fit. So this, the x-axis here is the proportion of rainbow trout admixture. So the pure westslope cutthroat trout are over here. The more rainbow trout alleles you have, the lower your reproductive output. So this is males and females. So as a general rule, it seems like hybrids are less fit based on this sort of data. But that's not the whole story. Here's another paper more recently from a bunch of people in this room, showing that this process of hybridization in these species is actually accelerated by climate change. So as the environment changes, the frequency of hybridization is increasing and will increase, and that will have effects on the native populations. Okay, so all of those sorts of analyses that I showed you depend on having some set of genetic markers. And ideally, if it's possible, it's nice to have diagnostic markers, which means markers that you know are fixed for one allele in one parental species and the other allele in the other parental species. So they're diagnostic and they tell you exactly which parental species they came from. So if we have a set of markers like that across the genome, it allows us to do things like estimate individual and population level admixture and put individuals on those x-axes, on those plots that I showed you. Potentially early detection of hybridization. If you have lots of markers, you have a higher sensitivity to be able to detect hybridization. And then also we can get into identifying particular loci that are showing interesting patterns and potentially responding to selection. So in cell lines, as you know, a lot of this stuff is complicated by genome duplication. I think maybe Jim will talk about that more. So it can be difficult to come up with these sorts of markers. But we used RAD, again, just to sort of illustrate how you can use RAD sequencing in different ways in these sorts of things. And our first attempt we had back in the old days, this was 2010 or something, we did relatively short read single-end RAD of samples from the parental species. We didn't have a reference genome, so we did de novo identification of the loci using stacks. But we did have some existing 454 sequence data that we could use. And we built a bunch of genotyping, SNP genotyping assays from that set of putative markers. One thing you can see is that if you just look at these numbers all the way from the top to the bottom, we did a lot of heavy filtering to get down to a set of markers that looked like they were diagnostic and well-behaved in all the sorts of ways we wanted. And then apparently something worked, because we had a high validation rate for that initial set of markers. But we wanted to expand that list. So then we did paired-end sequencing. We did contig assembly, like I talked about yesterday, using the paired-end data. we aligned the previously identified SNPs and looked at patterns of introgression. Skip forward so the basic idea here is let's see, we are right down here somewhere. So this is the North Fork of the Flathead River Basin. Rainbow trout were introduced here. We have samples from these populations and looked at patterns of introgression. One nice thing is that our estimates agreed pretty well with previous microsatellite estimates at the population level. And it follows this general picture of sort of introgression from rainbows flowing up into the river basin. So that the least admixed population is this one here that's farthest up. But we can also look for invasive alleles, just like I was saying, using this sort of outlier approach. So these are histograms of all of the different loci showing their frequency, relative frequency of rainbow trout alleles. And you can see there are a few outliers, and some of those outliers are common across these different populations. So those are loci that show much higher frequencies of rainbow trout. And then we wanted to make the set of diagnostic markers even better. So we did, again, paradigm sequencing of a wire sample. And this time we had a newly published rainbow trout reference genome that we used to align against. So here we are, here. These are where the putatively pure westslope cutthroat trout samples came from. And this was the reference genome that we now can use as a reference against which to align our data. And again, after various filtering steps, we got down to this list of diagnostic SNP markers. And now, since we have this reference genome, we can place those things on the map. Now you'll note, so now we have all these nice mapped diagnostic markers, and we can scan across the genome and look for patterns of introgression. One thing that you might notice, these are all the markers that align to the assembled chromosomes. These are all the markers that align to the essentially unassembled chromosomes. So even though a bunch of people published a paper saying we have assembled this reference genome, it's actually still in lots and lots of more or less unassembled parts. But it's still useful for what we're doing. And hopefully some of the people in this room can help make a better genome assembly. Okay, so that's the data that you're going to be looking at. Yep.
How problematic is this?
That's a really good question. So that's one thing that we haven't really talked about is the idea of reference bias, alignment bias. It is something that we've thought about and tried to deal with. The basic problem would be if alleles from one species preferentially align and alleles from the other species don't align, and you end up tossing out those reads. I don't know that there's a really easy solution, but we're definitely aware of that and looking for that problem to see if it's Okay, so the data that you're going to be dealing with come from this set of diagnostic markers. They've been genotyped in a bunch of other populations. And so what you have is the frequency of those diagnostic markers in a few other populations that are the result of hybridization between rainbows and native cutthroat trout. And I think I'll just turn it over to Ryan, who's going to sort of introduce the mechanics of how we do this long test of significance for these outliers.